All right, y'all, so this is our condensed intensive review of Unit 1.5, which is sensation and perception. So sensation is basically whenever sensory stimuli in the environment hits your sensory receptors. All right, so when there's light in the air, your eyes, your rods, and your cones pick up on that. If you're drinking something, there's stuff that's hitting your taste buds. All of that is sensation. And then when your brain makes sense of it, that's perception. Psychophysics is the study of perception. Let's see, pretty much all that what I just said. So we look at thresholds. So a threshold, in general, a threshold is like a doorway. But in the sense of sensation and perception, it's the minimum amount that you need to detect that a stimulus is there. So if it's totally dark, what's the minimum amount of light you need in order to be able to tell that there's light? There's a couple ways we could determine this. Some absolute thresholds were set by, I don't know who, because they're weird and random, but they only have to be accurate about 50% of the time, but random things like seeing a candle flame 30 miles away on a clear night, I don't think you have to know these. I think you just have to know in general what a absolute threshold is. The difference threshold is how much does that sensory stimuli have to change before you notice a difference? Like how many more lights have to be turned on before you notice it's a little bit brighter? And we call that the just noticeable difference or the difference threshold. Weber's law ties into the difference threshold. So if we're trying to figure out how much it takes, Weber's law says it's proportional to the intensity of the stimulus. So we talked about if the original intensity is low, like it's very dark or it's very quiet, you just need a little bit of light or a little bit of sound to notice that there's a difference. However, if it's really bright or really loud, then you need a significant change to notice the difference. We compared that with a full backpack versus an empty backpack. We also talked about the quiet, empty cafeteria versus the loud, full cafeteria. Let's see. Signal detection theory says that it's not just like a difference threshold like Weber's law, but that your experience, your expectations, motivation all tie into that. So, for example, if you're listening for the pizza man or woman, it could be a pizza woman driver, then you hear things better. Like you're more alert to signals. Or like a radiologist can detect cancer cells better than a normal person would because they're trained to detect those signals. Let's see. Subliminal perception, the idea that stimuli can be just below your conscious awareness. And you might be able to pick up on them. However, the effects are very minimal. I remember when we listened to the backmasking songs, hopefully nobody turned into a drug user or satanic person unless you were already that, and those are your choices. But hopefully those things didn't happen since then. All right, the Stroop effect, this is where we looked at these words and we tried to read them as fast as we could, and then we looked at these other ones where the colors don't match the word, and we tried to read them as fast as we could, but we tended to go slower. The reason for that is because of pre-attentive processing. So your brain wants to automatically process what it sees. So when it's reading where the color matches the word, it's easy. It's going very quickly. But when the color doesn't match the word, you have to slow down, and we call that attentive processing, where you're trying to slow down and consider only one part of the stimuli at a time. Selective attention is similar to this. It's the idea that you can focus your attention on one thing and tune out everything else. So we looked at some examples of awareness tests. There was a moonwalking gorilla, I think, and some other videos in there. The cocktail party phenomenon is the idea that if you were at a party or like in the cafeteria or somewhere, you have the ability to filter out other sounds and just focus on the person or a single conversation right next to you. So you can focus, filter, cocktail party phenomenon. Sensory adaptation is the idea that over time your senses get used to the stimuli around them and you really only notice changes. So that's why the swimming pool feels warmer after a while. It did not get warmer, you adapted to the cold water. If there's a stinky smell in the room, unless it's really stinky and you cannot adapt to it, eventually you just get used to it. So that's sensory adaptation. You don't feel your clothes on you all the time. Habituation is similar, however, you're doing it consciously. So, for example, if there's a loud sound outside and then you kind of tune it out after a while, but you know that it's still there, that's habituation, but you try to do it more consciously. That was part one. Part two is all about vision and our eyeballs. Some key parts of the eye. Let me just put them all up on here first. But we have our cornea, which is the outside window to the eye, and that's the one that's got all the sensory, like if you scratch it, it'll hurt. The pupil is the hole that the light comes through, and that one can change size based on the amount of light. That's the black part of your eye. The iris is the colorful part of your eye. And the muscle that controls the pupil. The lens focuses the light on the retina, and it changes that focus by bending its shape. The whole middle part of the eyeball is liquidy. That's the vitreous body. The back layer of the eyeball is the retina, and that's where the magic happens. The retina is where all of the sensory cells are located. The rods and cones line that. And then out here we've got where the optic nerve exits the eye and goes to the brain. We just said all those. All right, some other things, pretty much all that happens. Again, key term there, retina is where transduction happens. And remember that transduction is when it goes from being a light to a neural impulse that the brain is able to understand. Let's see, other things. The light travels all the way through the eyeball and it hits the retina, but then it comes back forward through the rods and cones. So here's where we have rods and cones. Remember that you have cones, which are good for color. They work best in daylight. You don't have as many of them, but they're very powerful because they don't share their connection. Rods you have more of, but they're for black and white vision, and they're in your peripheral vision. Let's see, other key terms. Okay, so here's where it is. So the light goes all the way to the back where it hits the rods and cones. Rods and cones are blue and red on here, and then it comes forward and it hits these bipolar cells and ganglion cells, and those are types of neurons. They're bipolar neurons. They have two axons, one on each end. And they do the transduction. It becomes a neural impulse there. Then their axons form the optic nerve, and that goes back here into our brain. So lots of magic happening in your eye all the time. It's pretty cool. Let's see. So the very specific spot for transduction is in the ganglion cells, and the ganglion cells are on the retina. So on the test, there's two ways they could ask, like, where does transduction happen? It could be in the bipolar ganglion cells, or which are part of the retina. So rods and cones, bipolar cells, ganglion cells, and then it goes out through the optic nerve, the optic chiasm, the thalamus, and the visual cortex. So the optic chiasm, I love this weird top of your head view, but behind your eyeballs, the optic nerves crisscross, and we call that the optic chiasm. So that's how visual signals get to the opposite side of your brain. Then they go back here to the thalamus, and then the thalamus is directing them to your occipital lobe at the very back of your brain. And the occipital lobe includes the visual cortex, which processes vision. Yay. Basically what I just said on that slide. So the full visual pathway, cornea, pupil, lens, then it goes through the vitreous body, then it hits the retina, then the signal goes through the rods and the cones, the bipolar cells, the ganglion cells, transduction or magic. Then we have a neural impulse that travels on the optic nerve to the optic chiasm where it crisscrosses to the other side of the brain. It hits the thalamus, which is our sensory relay system, and then it goes to the visual cortex in the occipital lobe. It's not likely they would ask you the whole visual pathway, but they might ask you about three things and ask you to put those three things in order. That was part 2. Part 3 is about processing vision. So a couple spots that deal with vision. Fovea is the central point in the retina. So it's about right here where I'm pointing. But that's where you have the most cones, so that's your best, sharpest vision. So we talked about if you're going to look at art, you want to look at it straight, head on, not out of the corner of your eye so you can see all the colors. The blind spot is right next to that, and that's where there are no rods and cones because that's where the optic nerve is leaving the eye, and there's a space there in the retina. Being nearsighted or farsighted has to do with the shape of your eyeballs, that if you're farsighted, you can see things far away, but close up is blurry. Nearsighted, you can see near objects close up, but far objects are blurry. Accommodation. So we know accommodation is a word that goes with language and schema development and cognition. But in vision, accommodation refers to the lens's ability to be flexible. So if you are looking at something close to you and then you quickly look at something far away, your lens is constantly reshaping itself so you can see things that are close and distant. So it's that reflexive action. It's just like blinking. Like you don't even have to think about it. Your eye is just constantly flexing that lens. That's accommodation. Pretty magic, all the things that happen in the eyes. All right, so when it comes to light waves, the hue helps us to determine the color. Wavelength is, I don't know, wavelength is the distance from the peak. And then the longer waves of red at the top, and then the wavelength determines what color it'll also be. Here it is. Different wavelengths of light result in different colors picked up by our retinas, and this is our color visual spectrum, just a small portion of the visual spectrum of what we as humans can actually see. Let's see, intensity, the amount of energy, and then when we talk about amplitude, that's how high the wave is, which we don't really think of much in vision, but in hearing, the amplitude is how loud the wave is, or how tall the loud wave. Hearing is louder if the wave is taller. All right, some other things, visual information processing. So parallel processing, the brain divides a visual scene into subdivisions such as color, depth, form. Let's see. I don't have to come back to this one because I don't really remember this one. It's new. We added it later. Let's see, some different theories about how color works. The trichromatic theory says that even though you only have 3 color receptors, red, green, and blue, in your eye for your cones, they mix together, and that's how you're able to see all the colors. So the mixing of those wavelengths of light help you to see all the colors. Color deficiency occurs when there's problems with a person's cones. There's a couple different types. Here they are. That's not it. There it is. Monochromatism and dichromatism. So with monochromatism, they are completely colorblind, which is pretty rare, but they have no functioning cones. And then with dichromatism, they only have 2 types of cones that are available to perceive color. And I don't think you need to know the specific types. Just know that monochromatic would be worse. Like you can only see black and white. Dichromatic, you can see some color. We looked at kind of the differences there. Opponent process theory is a theory that you see colors as the opposites, so red and green, yellow, blue, black and white, and that is supported by the idea of after images, where if you stared at these and you fatigue your rods and cones, you'll see the opposite colors. We saw the flag, and then we saw Jesus. Prosopagnosia is called face blindness, so it's where they have a random brain abnormality or brain damage, could be also, where they can see faces, but they can't connect them to anybody they know. Faces are basically like a blur to them. So instead, they recognize people by their hair and their voice and how they walk, and that's mostly it. Blind sight is the idea for people who are actually blind. They're able to pick up on some movement and facial expressions. It's just sort of a feeling that they have. Let's see, and that was it for part 3. Part 4 is hearing. So hearing depends on sound waves or vibrations in the air. We have hair cells that move in the cochlea, and that's where transduction occurs. Sound is measured in decibels. Too much sound, and that damages your ears. The highness or lowness of a sound is based on the frequency. Think of like an opera singer who's singing really high notes. It causes a lot of vibration. It could cause glass to break. Let's see, pitch. Or here it is. Timbre is the overall quality of a sound in this way that a person's voice, a guitar, or a piano could all be singing or playing the same note, but they sound differently, and that's because of the timbre. Amplitude is loudness, frequency is pitch, purity of the sound is timbre. So some structures for the ear. The pinna is the outer ear, and that helps to funnel sound waves down into the auditory canal. The auditory canal or the eardrum has hair in it, but those are not the hairs that we're talking about for transduction. We naturally produce wax, and that's to prevent dirt and things from getting further into the ear. The eardrum vibrates at the end of the auditory canal. It's also called the tympanic membrane. That vibration puts in motion the 3 bones, which are the ossicles, called the hammer, anvil, stirrup, also called the malleus, incus, and stapes. In there are the semicircular canals, which do not play a role in hearing. Those are for balance, but there's fluid in there, and that's how we have our vestibular system. The bones are vibrating against the cochlea. The cochlea opening is called the oval window. Inside the cochlea, that's where the hair cells are and the basilar membrane, and they're moving around like wheat in the wind or seaweed in the ocean, and that's where transduction occurs. And then coming out of there, we have the auditory nerve that goes to the brain. That's the same thing, opposite side. So our pathway of sound waves are funneled by the outer ear, which is also called the auricle. I forgot that earlier, or the pinna. And then those vibrations go to the eardrum, also called the tympanic membrane, causing it to vibrate. They hit the ossicles and the oval window. So ossicles are hammer, malleus, anvil, incus, stirrup, stapes. And then that puts vibration on the cochlea. And in the cochlea, the basilar membrane, transduction. Oh, in there is also called the organ of Corti and the hair cell receptors, and they make neural impulses. So here's the inside of that cochlea. So there's fluid constantly swishing around that, what do you call that shape? Snail shape. And that movement is what creates transduction. Organ of Corti is kind of like the lining of that. Let's see. All right, so then when you hear, it's going in your ear and it crosses over to the opposite side. It hits your thalamus, and then the thalamus directs those auditory signals to the temporal lobe, which is on the sides, and it hits the auditory cortex. So sound waves vibrate bones of the middle ear. Stirrup hits against the oval window, sets the fluid in motion. Hair cells are stimulated with the movement of the basilar membrane. Hair cells move against the organ of Corti, and transduction, the magic. The physical stimulation is converted into neural impulses. They travel along the auditory nerve to the thalamus and to the auditory cortex of the temporal lobe. All right, hearing problems. Different types. Noise-induced hearing loss damages the hair cells. Conduction deafness is when the vibrations are not being picked up by the tiny bones, so the eardrum is not vibrating enough. A hearing aid can help with that. Sensory neural deafness is where there's damage to either the auditory nerve or the cochlear implant, or to the cochlea. Repairs for that, sometimes cochlear implants work, and it does the transduction for the ear. There's some cochlear implants. Stimulation deafness is when you're exposed to very loud sounds. We talked about when you hear that ringing sound. That is the sound that indicates that hearing damage has occurred and hair cells are dying. Recently, research has shown that there may be some ways to regenerate hair cells, but that's still kind of new. All right, some theories of hearing or audition. The frequency theory says that the rate of nerve impulses traveling up the auditory nerve matches the frequency of a tone, and that's how we detect pitch, so the frequency of the vibration. Place theory says that we're able to figure out where sounds hit the basilar membrane, and that determines pitch. Kind of the outer edge of the basilar membrane is high pitches. So high pitches wear out your hearing first. So once you start to lose hearing, you can't hear high-pitched sounds. So if you're a grandpa, you can't hear sweet little granddaughter's voice as well because you have worn down the high-pitch hairs on your basilar membrane. Localization of sound. So we're able to detect that sound hits one ear faster than the other. So in this classroom, my voice hits your left ear slightly faster than your right ear. So that helps you to know or to localize that the sound is coming from the left side of the room. Whew, that was all for hearing. Part 5 is the other senses. So smell and taste are known as chemical senses. We've got the gustatory system, which is our sense of system for taste, like gustatory. And then olfactory is smell. Think the smelly olfactory. Recall that smell does not go through the thalamus, but all the other ones do. You do not have to know the pathway for the other senses like you do for vision and hearing. Here is the surface of the tongue. The taste buds are located down there in the grooves. When you look at your tongue, you cannot actually see your taste buds. Instead, you see the papillae. The papillae are bumps, and in between those bumps, that's where your taste buds actually are located. And down there in that taste bud, that's where transduction occurs. Let's see, but it does smell, goes through the olfactory cilia, so there's hair cells up in your nose, and transduction occurs there. It goes to the frontal temporal lobe. Let's see, things about how do we get flavor. Flavor is a result of sensory interaction between both smell and taste, but also like touch sensations in your mouth. The taste sensations that we know are bitter, sour, salty, sweet, and umami. There was another one we learned. I can't remember what it is right now. It was like, I don't remember, but it's in here. I'll have to come back to it later. I don't know if it's in here. And then flavor also depends on the temperature. Think like hot cheese versus cold cheese, like melted cheese. Odor, texture, and taste. So all of those contribute to flavor. There was another word. I'm going to have to, hopefully it's in here. They used to think that the tongue was divided by like sensations like that, but now you can taste all the flavors all around your mouth and the roof of your mouth and the top of your throat. So there's all those. Let's see. Most of our taste sensations are learned preferences, some of which started while you were still in the womb. Super tasters versus non-tasters. Super tasters have way more taste buds than normal, and that's why they're able to detect flavors more intensively. Statistically, Asian women are most likely to be super and white males least likely. Evolutionary perspective says that being a super taster was so that as a gatherer, you could detect if foods were bitter and that they could be poisonous. All right, skin. So our touch sensation, the skin is the largest sensory organ. So the largest organ in your body too. So not your intestines, your skin is an organ. Touch, pressure, warmth, cold, pain, fine touch. All of that is processed by the somatosensory cortex, which is in your parietal lobe at the top of your brain. When it comes to feeling pain, the gate control theory says that you have this hypothetical gate at the top of your spinal cord, and you can open and close that gate to divert pain. So when you're distracted, you can close the gate. So things like rubbing your elbow will help block pain, or thinking about something else, or talking to somebody while something painful is happening. Gate control theory says you can help block it out the pain that way, within limits. There's also something else that talks about that, there's a fast or a slow pathway. So some pain goes very quickly, and a lot of that is processed as reflexes through the spinal cord, and then other pain goes more slowly up the spinal cord to the brain. Other senses, your kinesthetic sense is position and motion. Your vestibular sense is your body's balance. But kinesthetic sense is like what lets you to walk down the stairs while you're texting, which is real dangerous. Don't do that. But if you have really good kinesthetic sense, you could probably do it. Vestibular sense is what causes you to be dizzy. It's controlled by the fluid in your ears in the semicircular canals. Oxytocin, that was on the mock exam, and some people forgot it. Oxytocin is the hormone that is released with the sense of touch. So we talked about 20-second hugs, and how 20-second hugs feel good because you get a release of both dopamine and oxytocin. Oxytocin is also released as like the bonding hormone when mothers nurse their babies when they're breastfeeding, both from the touch of holding them and just their body releases that chemical along with the milk. Part 6, perception and gestalt. So again, perception is about how your brain makes sense of all of that information, especially when that information is incomplete. Synesthesia is a weird neurological condition where your sensory pathways they're mixed up. So for example, you taste colors, or I've been watching Love on the Spectrum, and one of the guys on there, every color he sees is a number to him. In his mind, it comes up as a number. So that would be synesthesia. So the girl in the video is like, this is yellow. That's number 3 for you, right? He's like, yep, it's number 3. Whenever they come across a new color, she asks him like, what number is it? And it's a different number for him. So synesthesia. All right. Perceptual set is the idea that your experiences and kind of things things that are going on in your mind affect what you perceive. So that's why the image there could look like a couple different things. We talked about backmasking when we listened to those songs where they were played backwards, that you couldn't really hear anything, but if you read on the screen at the same time what the message was supposed to be, then you could hear it. That would be perceptual set. So predisposition causes you to perceive things a certain way. All right, but perception is an active process, which is why you can kind of change what you perceive. Gestalt is the idea that our brain wants to see the whole and not just parts. So it fills in the gaps. Figure ground is the first Gestalt principle. That's the one that says we can change whether we're looking at the foreground or the background. So in all of these pictures, you can see two different things because you're changing figure and ground. Some more examples. Proximity says that we perceive numbers and objects that are similar as a group. I'm going to go through these real quick. Similarity, we see objects that are similar in color easier. Continuity, simplicity, closure. Perceptual inference says that we fill in the gaps with what our senses don't tell us. So we know that this road is going to keep going even if we can't see it there. Maybe you kept your Gestalt name tag where you used all those principles to make your name or some other word. All right, monocular and binocular cues. I think this is the last section. So depth perception is the result of either binocular or monocular cues. Monocular cues use only one eye. So even if you only had one eye, you could detect depth when you're looking at pictures. Some examples of monocular cues: linear perspective, where parallel lines converge. Texture gradient, stuff that's farther away is less clear. Interposition, which is the overlapping of objects, helps you to see depth. Relative size, where things that, how close they are, they appear larger. Like I look like I'm the same size as the Leaning Tower of Pisa, but in reality, I'm just closer to the camera. That's called forced perspective. Motion parallax is where objects that are in the distance look like they're moving slower or the opposite direction. Binocular cues are using both eyes. So retinal disparity is the difference between what each of your eyeballs sees, and then convergence is how much your eyeballs turn inward as objects get closer to your face. Some of the optical illusions, I don't know if these are going to be on there. The Muller-Lyer illusion, these lines are all the same, but because of the arrowheads, they look different. The moon illusion, the moon never changes size, but it looks bigger near the horizon, and that's because you're able to compare it to on-Earth objects. The Ames room, these guys look like this guy's way taller and this guy's way shorter, but the room is irregular. The ceiling slopes down, the floor slopes up, the room is shaped kind of like a trapezoid. Let's see. Bottom-up processing, I'll get to those at the end. Perceptual constancies, there was one or two questions of this on the mock exam also. But size, shape, brightness, sometimes those look different in pictures, but in real life, they remain constant. So for example, a door is always a rectangle. Even if you perceive the door open and it looks like a different shape, like a trapezoid, it is still, in reality, constantly keeping its shape of rectangle. This guy's green pants are still green in reality, even though in darker light, it looks like the pants change color. The pants do not change color, but your perception of them do. Let's see, perceptual set, that was an earlier one. Okay, top down, bottom up, and top down processing. So bottom up is where we see the parts and then we see the whole. So if you started looking at a person from their feet, you wouldn't know who they are. You start at the bottom, you see their feet, their knees, their hips, and then finally when you see the whole person, then you know who they are. So that would be like tasting individual flavors, hearing individual notes in a melody. Top down is when you see the whole thing and then you can kind of zoom in. So like you go to the cafeteria and you're like, wow, I can see all these people. But then if you focus in, you could see individuals. So being able to see that this is an image of a cat would be top down because you're seeing the whole image and not just the spots. Same thing with this type of art. Top down, trying to read bad handwriting, or things like this where the words don't make sense, but if you slow down, you can read it because your brain is seeing the whole word and not just the individuals. That's the whole section. Yay. Let me hit pause.